Hello, thank you for coming to this education course. I revised the presentation title to human-brain mapping of individual differences, reproducibility, replicability, and reliability to reflect the contents more appropriately. During this course, you will learn about the background of reproducibility challenges and the basic concepts of reproducibility and reliability. I will take subcortical laterality as a working example to show you the two concepts. Coming after the two fundamental concepts, I will introduce the measurement theory and the definitions of reliability and validity in the perspective of studying individual differences. Accordingly, I will bring you the demonstrations of how we measure individual differences in subcortical volumes and their reliabilities. The reproducibility challenge has been raised for almost every field of experimental sciences such as medicine, neuroscience, psychology, etc. Scientists have summarized the threats to reproducible science, including failure to control for bias, low statistical power, poor quality control, p-hacking, and publication bias. These issues are sitting in different stages of scientific research, including generate and specify hypotheses, design the study, conduct the study, and collect the data, analyze the data and test the hypothesis, interpret the results, and publish them. A manifesto for reproducible science has been proposed in 2017 to handle these threats with pre-registration, team, and open science practice integrated into the different stages of the scientific research. Patel and colleagues developed a protocol to represent the 11 components of a scientific study: population, question, hypothesis, experimental design, analysis plan, and data, experimenter, analyst, code, estimate, and claim. The first 5 components are in the intent of a study, while the 6 remaining components are in the conduct of the study. Based on these components, we define reproducibility as reperforming the same analysis with the same code using a different analyst, and define replicability as reperforming the experiment and collecting new data. In the next slide, I will leave 1 minute for you guys to finish the quiz. Okay, I think you guys already got the answer. They are all, the answers are in the bottom of the this slide. So before we get into the demonstration of the reproducibility and replicability, I introduce two open resources for human brain mapping of the individual differences. The first one is an open resource for reproducible, replicable, and reliable brain research and imaging neuroscience, so-called 3R brain. It collected both brain imaging and behavior data from 103 healthy young adults using an experiment designed for repeated measurements. Each participant received 5 MRI scans at 3 different scanners. Specifically for each of the two 3 Tesla scanners, GE 750 and Siemens Prisma, a 2-week test-retest design was applied to each subject who had the last MRI scans at the Siemens 7 Tesla scanner. The entire data collection was completed in 130 weeks. The multimodal MRI measured individual level features of both brain morphology, T1 and T2, structure, DTI, and function, both resting state and natural stimulus functional MRI. Each scanner implemented the best performance of these imaging sequences. Beyond the multimodal imaging measurements, questionnaires, physiological tests, and behavioral tests were also employed to collect the corresponding behavior data. The second is FEMUR's Young Adults Human Connectome project. One nice feature of HCP data is the pre-processed data publicly available for you. I would not go in too much details, and you can get its every piece from the website. Now, let's demonstrate the reproducibility and the replicability of asymmetry of subcortical volumes. Many studies have reported the findings of lateralization in subcortical regions. However, the replications of these findings are quite limited. By now, the largest study is conducted by Enigma using MRI images of more than 15,000 subjects from 52 sites. It reported that each subcortical region exhibited some degree of asymmetry, while pallidum and putamen showed such effects on the asymmetry. Here, I will show you the reproducibility and replicability of a scientific study on the subcortical asymmetry using both 3R brain and HCP data. So here is the 11 components of the original study. The population is young adults. We try to reduce the variability of age. The question we are asking is, are these subcortical volumes different between hemispheres? So we hypothesize that the subcortical volumes are different between the two hemispheres. So the experimental design we use is for 82 healthy adults, including 44 males of ages between 18 and 36 years old. We use the morphological MRI measurements. We're going to analyze the data by using subcortical segmentation tools, focus on the symmetry index. The statistics we use is the paired or two sample t-test. The data from the data is structural MRI images from GE750 scanner. The experimenter is including the research assistant team and the brain imaging operation team. The analyst is Dr. Yinshan Wang, who is a postdoc researcher from my lab. The code will be written in R and in R scripts. The estimates are from the paired and two sample t-tests. The answer and the conclusion to the scientific question will be statistically corrected by using the Bavarian correction. Specifically, we employed the online brain segmentation tool, WallBrain, to extract the volumetric measures of the 7 subcortical regions. So this slide gives you an overall picture of the original study. The analyst is Dr. Yinshan Wang, who compared volume differences between the left and the right subcortical regions using the pair t-tests, as well as the asymmetry differences between females and males using two sample t-tests. He claimed that the hippocampus was rightward, while the thalamus and accumbens were leftward. The effect size are plotted in the left, and the statistical power levels are valued in the color boxes. So in the reproduced study, Yinshan sent all the data and the codes in R. These are R scripts to another analyst, Zhu Qinggong, who ran all the codes from her side and reported a fully reproduced version of the original study. It indicated that the different versions of both hardware and software seem no effects on the study. I took all the data from Yinshan and analyzed the data using my own code, which is in MATLAB scripts, and reported almost identical findings except for the changes of statistical power. Yinshan then analyzed the new data of the same group of subjects collected from another scanner. Now we can see the statistical estimates changed, especially for the effect sizes and the statistical power levels. But the claim remains. So Yinshan again analyzed another new data of a subset of these subjects, only 36 subjects, from the 7 Tesla scanner. The results changed and claimed more regions with asymmetry. The asymmetry oh, sorry, I think I missed this part. So this part is Yinshan analyzed the new data from a subset of these subjects, but the data obtained from the 7 Tesla scanner. So the results changed and also claimed more regions to show asymmetry. Especially the asymmetry of thalamus seems not detectable under these replication conditions. I analyzed the data in replication study 2, and again basically replicated the original study, although the estimates of statistical power changed. This is also true for the replication study 3. In replication study 6, I tested the scientific hypothesis by using the HCP data from the 100 unrelated subjects. We can see somehow dramatic changes for all the statistical estimates and even the claims. For example, occupants was leftward in the original study, but rightward in this replication study. This can be further confirmed using the entire 1,113 subjects data from the HCP 1,200 release. It demonstrated that all the subcortical regions are asymmetric. It is very interesting that sex-related asymmetry was consistently reported no effects across all the replication studies I present here. Coming to the end of this part, it seems hippocampus and accumbens shows reproducible and replicable asymmetry, although the accumbens demonstrate the opposite direction between Chinese and Western samples. Taken together with those observations offered by the large-scale Enigma literature studies, I would conclude that all the challenges of the presented replication studies very likely reflect the measurement variabilities of individual differences related to age, sex, scanner, magnet strength, culture, etc. In building a sense of individual differences, measurement theory is fundamental to understand the variabilities related to measurement targets, tools, and metrics. 1,000 functional connect project represents a very nice example to understand the potential sources of the measurement variabilities in mapping individual differences in human brain function, such as center, age, and sex. These variabilities can be categorized into 3 different types, including those of disorder or trait related, the contaminants such as head motion, and random errors. The first 2 parts are variabilities between subjects, while the random errors are within subject variability, which must be measured with repeated experimental design. According to the 2 equations I listed here, we can understand reliability as a form of replicability for studying measurement vulnerabilities. That is the replicability of between subject vulnerability across different measuring occasions. It is within subject vulnerability. This slide demonstrates the anatomy of reliability. It indicates that higher reliability requires larger between subject vulnerability and smaller within subject vulnerability. That means reliable measurement can recognize one person from another more easily, while more stable across random noise conditions. A clinical measurement requires the almost perfect reliability greater than 80%. We never know the true effect size to be investigated, but we can observe its experimental version by using some measurements developed. In theory, the observed effect sizes are their true effect sizes weighted by the measurement reliability. Here, I'm showing you the plots of interactions between reliability, sample, and effect size for a given statistical power and paired t-tests and two sample t-tests. It clearly shows that given an effect size, a study with a more reliable measure needs less samples. Given measurements with the almost perfect reliability, 300 samples can offer a study of 80% power to detect the small effect sizes. Given measures with the slight reliability at less than 20%, big data are required for a study to detect small effect sizes. So now let's play with the 3R brain samples. Just to recall, it offers the repeated measurements from the 3 Tesla scanners, which I plotted, I put into the blue box. We use linear mixed effect model to measure various different variabilities. These variabilities can be related to subjects, imaging sessions, and imaging size. Also interactions between different sites and different subjects. Interactions between different sessions and different subjects. So interactions between sites and sessions. So based on that model, we can define the overall, the inter-site, and also inter-session, also called test-retest reliability, based on these variability components. By fitting the model into the 3R brain data, we map out the between subject variability, it also called individual differences, between site and between session variabilities, as well as the volume measurement reliability of the 7 subcortical regions. it shows that the almost perfect reliable volume measurements are in thalamus, putamen, and caudate. So this slide is for the same findings for test-retest reliability. We can clearly see the test-retest reliability is much higher than the overall reliability. How about the intersite reliability? So this is the intersite reliability. So it is much lower than intersession reliability. So all the variabilities and the reliabilities can be also estimated for the asymmetry index of the 7 subcortical regions. The slight to fair reliability of the equivalence symmetry measurement has very strong contrast to the substantial to almost perfect reliability of the hippocampus measurement. This may help us to understand the previous observation of their performance we had from the reproducibility and the replicability demonstrations in the second part of this course. So to summarize, a standard for 3R terms was introduced in this course. Measurement reliability is fundamental for science of individual differences. Measurement reliability should be carefully evaluated and improved into a standard for translational studies. Open science should focus on building rich datasets for measurement reliability and validity research. So all the codes, all the data and codes present in this education course are accessible in this website. And for you to further reading, I recommended the 3 papers posted here, posted in the right. So this is all the education course content I'm going to present to you. Thank you for listening.
Hello, it's very nice to be here, virtually, but here for this presentation. And I'd like to thank the educational committee and the program committee to allow us to present this interesting workshop on population neuroimaging, how to responsibly handle big data in the age of biobanks. I'm Tonya White. I'm an associate professor at the Erasmus University Medical Center in Rotterdam, the Netherlands. I'm director of the neuroimaging component of the Generation R study, which is a large population-based imaging study. I'm going to briefly introduce the rest of the speakers. Henning Tiemeier is professor at Harvard University. He has 20% time here, and over the past decade, he's been one of my closest collaborators. He's going to speak on the 7 plagues of conventional brain imaging studies towards population neuroscience. Ryan Muetzel is a young, talented assistant professor here. He was my PhD student and then did a postdoc with Henning and I here at the Erasmus University. And he co-directs the neuroimaging component of the Generation R study, and he's going to be talking about application of epidemiological concepts in neuroimaging. Beckmann is a professor at the Donders Institute in Nijmegen. He's well known in the neuroimaging community, probably needs no introduction. And he is going to be speaking on big data for precision neuroscience, novel approaches for charting resting state functional connectivity and connectopes. Anqi is an associate professor at the National University of Singapore. And she actually is, to some extent, my counterpart for the Gusto study, which is also a large population-based study in which they scan infants, which is very challenging. She is going to be talking about population neuroimaging, meets genetics, important considerations with data, both big and small. And then last but not least is Xi-Nian Zhao. He was at the Chinese Academy of Sciences, but he's moved across town to Beijing Normal University. And he's a professor of population neuroimaging, and he's going to be talking on the 3 R's of reliability, reproducibility, and replicability. So one of my goals in my talk is to kind of lay the foundation of population neuroimaging. I'm going to talk specifically about some important concepts related to selection bias, but really to describe some of the issues. And at the very start, I'm going to talk about the very first imaging study in epidemiology, but it wasn't a brain imaging study, but rather an imaging study following an cholera epidemic, and take you back now to London in 1854, August 31st, 1854. So Abraham Lincoln was president of the United States. They were in the midst of a civil war at the time. And cholera in the Soho district of London, there was a cholera outbreak in which 127 people died within 3 days, and upwards of 500 people. And this is John Snow here, and he had a hypothesis surrounding wells. So these are where people got their water in London at the time. And he mapped out where the deaths were, and eventually went to the local authorities and said, presented his data, and he said, there's something going on with this well. I think that's where the cholera is coming from. It was later tested, and in fact, they had the handle removed from the Broad Street pump on September 8th, 1854. You can see that the rates were already declining then, but they did later find out that it was a diaper that was from a baby that had died that was seeping into the well. It was not discarded in the well, but it was seeping into the well. So this is the very first imaging study in epidemiology. But now we go to what is population neuroimaging. And actually, epidemiology is in the news a lot now with the COVID-19 pandemic. I guess if it were today, epidemiology would be called pandemiology. But it's this intersection between epidemiology and neuroimaging. And epidemiology focuses much on environmental determinants. And so genetics, if included here, it covers a more complete picture of this relationship. But actually, the population neuroimaging covers this intersection here. The definition of epidemiology from the disease control is the study of the distribution and determinants of health-related states or events in specified populations, and the application of this study to the control of health problems. And we're having a virtual meeting at this time because of the pandemic. So you can think this is the time when you think of the distribution and determinants of the COVID-19 in specific populations, which in this case is worldwide, and then control of health problems in terms of flattening the curve versus other countries use different techniques. But this is a definition for epidemiology. Neuroimaging is something we're all very much used to. It's the use of various modalities to directly or indirectly image or map the structure, function of neurochemistry of the central nervous system. If you take an intersection of these two definitions, then what you get is population neuroimaging, which is the use of various neuroimaging modalities to study the distribution and determinants of health-related states or events in specified populations, and then the application. But I want to focus right now on some specific words within this definition. And I'm going to start with specified populations, because there's one thing that epidemiology, there's many things that epidemiology can help in terms of improving neuroimaging studies. One of them is to really understand this notion of population. So I'm a pediatric neuroimager, so I'm going to discuss different domains in which some of the large pediatric neuroimaging cohorts fall into. So, and this is the population that they're drawn from. So for example, typical development cohorts, generally they involve individuals who don't have any psychiatric disorders, are not cognitively impaired, and they're following just more of a typical developmental trajectory. So you're excluding people in order to have this typical development population. Clinical and high risk are generally focused on specific clinical. So for example, ABIDE 1 and 2 focus on autism. The ADHD 200 focuses on ADHD, and CANDA focuses on alcohol, but they focus on either a clinical disorder in which they have patients and controls, or high-risk groups. And high risk can be both genetic at risk, such as family members who have the specific disorder that you're interested in studying, or behaviorally at risk in children or adolescents or adults who have symptoms of the disorder, but do not meet the full diagnosis. I'm going to be focusing a lot on this population-based group here. And there's a number of different large population-based studies, and I'll be describing how these are quite different. What they do typically is they try and capture representation of a certain population. So for example, for adults, there's the UK Biobank, which they try and capture the population characteristics within the UK. So I'm gonna, there's two studies here, and we have representation from these two studies, Generation R and the Gusto study in Singapore. These are actually studies which children have been scanned and their birth cohorts. So the recruitment took place during prenatal or early life, and they've been scanned during children later. And there's some advantages of birth cohorts because actually you're starting very early. There can be bias because of people falling out, which is called attrition bias. But let me talk a little bit about the design of the Generation R study, because it gives an idea of one approach for a population-based study. And that was pregnant mothers who had a delivery date between 2002 and 2006, who lived within these zip codes here, shown in blue, were invited. And that was the only inclusion criteria. So everyone, regardless of cognitive level of functioning, psychiatric disorder, everyone was, all mothers who were pregnant, essentially within these zip codes, were invited, and about 60% decided to participate in the study. So given this example, there are, and this is one of many large population-based cohorts, there are advantages to these large population-based cohorts, because you're drawing from a broader population. You can better estimate the prevalent rates and distribution of specific variables in the study. So when we look at, for example, the Generation R study, and you look at ethnicity, the total sample was close to 10,000. You can see that that less than 50% were Dutch individuals, and that's a representation. It's representative more of Rotterdam than the entire Dutch population. And Henning is going to be talking more about generalizability and representation, but it's quite representative of actually of Rotterdam. But then we had wave 1 and wave 2 of our neuroimaging waves, and you can see that the distribution changes between these. It's not quite the same as here. Wave 1 had some oversampling, and wave 2 we tried to recruit as many as possible, and but still it's more reflective of the original. But there's some differences. But the closer you get to the general population, the more you can make inferences towards the population that you're studying. So the other advantage to large population-based cohorts is you can capture symptom dimensions across the spectrum. So when you look at more cases versus controls, so say you have a psychiatric diagnosis here, and then you study a group of individuals who are, this is here a continuous measure of clinical symptoms, for example. You're recruiting populations to match a DSM diagnosis versus a specific control. Let's say here's some neurobiological measurement. It could be modularity of brain connectivity. It could be fractional anisotropy in specific regions of the brain. And here's a continuous measure of clinical symptoms. If you draw from a population, you get a much more distribution, a broader distribution of clinical symptoms, including those that might fall into this area, which are subclinical traits. And then you can ask questions such as, is there a continuous relationship between the neurobiological measurement and the clinical symptoms? So if you have more ADHD, do you have more ADHD symptoms, inattention symptoms? Do you have more differences in your caudate, for example? Or is it such that you have a flat line, and once you get to a certain threshold of diagnosis, then you start seeing the differences in your neurobiological measurements? So these are things that can be tested in these large population-based studies. Other advantages to these large population-based cohorts is you can capture brain changes related to the emergence of symptoms or disorders. The large study, big data, gives you more power to test relationships, and it provides opportunities to assess unbiased. No study is really unbiased, so it's better to say less biased relationship between variables. So you'll see here that bias shows up a fair amount. And so I'm going to talk about the different types of bias that are present. So you can have a selection bias, in which individuals are more likely to be selected than others based on certain traits. For example, single mothers may have it be so busy that they're less likely to participate. Reporting bias is something that we're all familiar with. It's easier to publish positive findings than negative findings, which results in some level of reporting bias. Or some people may not want to talk about certain characteristics of themselves as opposed to others. There's analytical bias, which is the bias in the way data is analyzed, and it's not necessarily covert. It can also be overt or just differences in the way data is analyzed. I think of Mark Twain's quote, "Lies, damn lies, and statistics." But this bias does not need to be conscious, or it can be just differences in how the data is analyzed. Exclusion bias, which, if you can think about people with metal in their bodies, can't go into MRI scanner, that would be a certain exclusion bias. They're excluded for different reasons. Language, other factors can result in certain populations being excluded. Attrition bias is problematic in longitudinal studies. There's a loss of participants that occurs in a non-random fashion, and oftentimes the individuals that you most want to study are the ones that unfortunately are most likely to drop out of the study. At least for some psychiatric disorders, that's true. Hopefully this is something of the past, the funding bias, but I'm sure it still happens. A selection of important variables that favor the financial sponsor of the study. And then there's a career enhancement bias, which is playing with data until you come up with a positive result, which is also called p-hacking, which is unethical and should not be done. So let me talk about one specific example of selection bias, which is called Bergson's fallacy, which was reported in the 1950s. And Bergson did it using two different conditions. So people who came to the hospital had either condition A and condition B. And he showed that if you restrict the population, so in this case, he restricted it to the population, it can cause a negative correlation between different symptoms between condition A and condition B. And I'm going to show you this more specifically and then show you a simulation of this. So essentially, if you have your entire general population here and you have condition A and condition B, and you have some overlap that could happen, it could be independent, it becomes correlated if you remove the entire population and just look at A and B. So let's look at a simulation of this. So let's say you have a clinic that recruits children with either ADHD or ASD. And you look at 1,000 children with symptoms with ADHD and ASD. Now, what I did for this simulation, and this is something that you can do for simulations, is I made it uncorrelated, or the symptoms for each individual had a measure of ADHD and a measure of ADHD, and I made those so they were not correlated, and through a random number generator. I also transformed it so the symptoms looked more like what you see in specific checklists or lists of symptoms, where you have no symptoms at 0 up to a certain number of ADHD symptoms. So these two graphs refer 1,000 children, the same children, but one of them reflects their ADHD symptoms and one reflects the autism symptoms, just essentially from the general population. So what I do now is I begin with the whole population, and then I slide a threshold. So a threshold, people don't come to the clinic if they have no symptoms. So what I do is I say either you have ADHD symptoms or autistic symptoms, and then you come to the clinic, starting with the general population. And then you look at the correlation, and you can see in this simulation that this is the threshold. So 0 means I include everyone in the entire population, and I did 50 iterations for each symptom threshold. There's no correlation, which is from the entire population. But the more that you say either you have ADHD or you have ASD, come to the clinic, it becomes negatively correlated. If your clinic is ADHD only, then it remains uncorrelated. But it's just, if you're looking at a specific population restricted to 2 different disorders, then it can cause a certain bias between those disorders. Now I'm going to look at another example of selection bias, or possible selection bias. I'll let you think about it. So I want you to focus on, this is a study, this is actually my study from 2006 in biological psychiatry, where we had adolescent and patients and controls with schizophrenia compared with adult patients and adult controls with schizophrenia. And this is the demographic table. And I want to focus on the IQ, because you can see that the adolescent patients and the adolescent controls have a 20 to 25 point difference in their IQ, and adults a little bit less, but about the same. So you might say, wow, you're comparing apples and oranges here. They should be matched on IQ. But it's not so easy, because with schizophrenia, the emergence of schizophrenia is also related itself to cognitive decline. It's part of the illness. And if you look at the father, the educational level of the father and mother, you can see that these are actually quite well matched. In fact, the adult patients' parents have higher educational level than the adult control subjects. So, and if you think that educational level is related to offspring IQ, which is probably true, you could say, well, maybe this is more the result of the illness itself. But you couldn't get that unless you show all the information to let the readers make the decision. So one key point here is to provide enough information so people who are reading your papers can also look at the level of potential selection bias. And actually presenting both with and without controlling for IQ could be helpful in this case. So I'm going to move on back to the definition, but this time we're going to look at other terms in this definition, the determinants of health-related states or events. And for this, I'm going to turn to the UK biobank study. So this was a paper by Steve Smith and Tom Nichols, published in 2018 in Neuron, in which they present all the univariate, 14 million univariate analyses of the different non-imaging variables from the UK biobank study with these imaging metrics, showing that actually 100,000 pass FDR correction and 15,000 of those pass Bonferroni correction. So you could say the future is going to be full with a lot of different papers coming from the UK biobank, which is true, but I want to go into some of the issues related to this because we've actually been looking at this issue. So if this were genetics, this would be a much different question, because if you're far enough away related to linkage disequilibrium, if you're far enough away, then they're independent. But with these type variables of lifestyle, exercise, work, physical, cardiac, these co-vary with each other, and that makes it different in terms of how you look at variables, in terms of looking at relationships between variables. This is an example, not 14 million univariate analyses, but this is 990 univariate analyses from a study we're doing on an environmental wide association of autism spectrum disorders. And you can see the univariate analyses not controlling for anything also has a large number, the purple line here is Bonferroni correction, a large number of significant variables. Those aren't significant, or they're significant in the test, but if you consider a variable such as sex, so we know that autistic symptoms or autism is 4-to-1 male to female ratio. So any variable that might be related to sex, or any variable that might be related to a certain health measure, that may co-vary with others. And then you see the inflated significant findings. And here's an example of that, of difference. So education level mother, so it took a number of variables. So here's how does education level mother co-vary with all the other 990 variables, age, mother at intake. You can see that unlike a map of what genetics would look like, you get high correlations between different variables. And variables can be considered mediators, moderators, confounders, or covariates. And Henning is going to go into this more in depth, but you can have a certain exposure or condition. A mediator means it's in the pathway. Oftentimes it's thought to be a causal pathway. A moderator means it affects it can affect the estimate between the independent and the dependent variable. Confounder means that it can affect both the exposure and condition and the outcome, and covariate is mainly affecting just one, and confounder is actually also a covariate, but a subset. So if we take an example here, so you have smoking. So let's say you start doing a study within the UK biobank and you have smoking versus hippocampal volume. Head movement and MRI. So head movement, I'm assuming it's not related to long-term smoking, because possibly it could be, but so head movement I put in as a covariate, as potential covariate there. Cardiovascular changes, now that potentially you have smoking, hippocampal volume, that could be mediated by cardiovascular changes. So that could be in the pathway between smoking and, let's say, lower hippocampal volume. What about socioeconomic status? So socioeconomic status could potentially affect both hippocampal volume And people from lower SES tend to smoke more, so an SES would be more likely to be considered a confounding. I don't have COVID, hope not. And then ApoE carrier, well, there's evidence that from a large meta-analyses that can potentially be an effect modifier. It wouldn't be in the pathway because your ApoE status does not, smoking does not cause your ApoE status. But there's many variables to consider: sex, pack years, educational level, activity level, lifestyle factors, alcohol. It's actually, how do you consider each of these variables? These come both from the literature and making decisions, but also there's ways to test specific variables to see if they affect or not. So finally, I'm going to end with just an overview, or just to mention there's a COBITUS document. One of the things that is discussed is the study design section, and this has a lot of information for the best practices and for reporting. Reproducibility is spelled wrong. I hope you don't use that in a reproducible way. But in any case, this document can be very helpful to describing some of the things, including what to report, so that people can understand what inherent biases may be in the study. And then also in the COBITUS report is data sharing. So this is my plug for data sharing, and I will end with the possibility of virtual questions. So thank you very much for the opportunity to present, and email me or find me if you have any specific questions. Thank you.
Hello, my name is Ryan Muetzel. Together with Tonya, I lead the Generation R imaging initiative in Rotterdam at the Department of Child and Adolescent Psychiatry, Erasmus MC. We are very grateful to OHBM, to the OHBM Educational Committee, for giving us this opportunity to organize this session, which we feel is increasingly important for the brain mapping community. As you heard from Henning, in this talk, I'll be following up on some core epidemiological concepts which we feel are important enough to warrant a bit of repetition and additional detail. Let's see. There we go. I have no disclosures to report here. Just so everyone knows where this is going, I'll begin with a bit of background on effect sizes, confounding, and how these things are related. I will then go into some details on specific example of whole brain exploratory analyses to illustrate why we need to take confounding seriously, even if our tools don't fully allow for it. Next, I'll provide some info on selection bias, and then I want to finally wrap up with something to chew on over adjusting your brain imaging analyses for total brain volume. I hope by the end of this talk, you'll be able to identify confounding in your studies, and we'll have a better grasp of the various consequences of confounding for your brain mapping research. I also hope that you will be able to identify selection bias in your work, and will also be able to critique it when reviewing papers in brain mapping. Lastly, as I mentioned, I hope I can get you to reconsider a standard practice in morphological brain imaging of blindly adjusting for total head size. More on that later. To kick things off, for those of you who are looking for a bit of introductory reading material on population neuroscience, which you may have already noticed from the other talks in this series, a really good starting point is a paper by Steve Smith and Tom Nichols, which is used throughout this series as a discussion point. Another paper by Thomas Paus on population neuroscience is worth a read. And coming out this fall, a little plug for Henning and I, is a chapter that we put together on population neuroscience that's coming out in a book on child and adolescent mental health. Now, Steve Smith and Tom Nichols discuss effect sizes in several ways in their paper. One important way that this is highlighted is that with larger and larger studies, we're able to detect smaller effect sizes. And what do I mean by effect size? Well, this can take on any number of forms, R-squared or explained variance, for example, measures of association, such as standardized regression coefficients, mean differences, etc. Everyone has their own statistical technique to measure their particular research question. This problem, however, is nicely highlighted by Steve Smith and Tom Nichols in their paper, where using the UK Biobank, they ran something like 14 million tests and then bound-firmly corrected those tests. Many significant associations remained at much less than 1% variance, which is really small amount of explained variance, small associations. The general conclusion from this in their paper is that with very, very small effect sizes, at least on their own, they are likely not always so meaningful. There's more sophisticated methods for pooling or, say, aggregating these effects are needed. So if you think about multivariate techniques, machine learning, and so on. Other methods are also available, and I'm happy to share that these will be discussed in detail by Christian Beckmann in another talk within this series later on. But so there you are, with small effect sizes, many may not be so meaningful, and we need to treat them differently than we do with larger effect sizes. But what about large effect sizes? In brain mapping, we're very often focused on p-values, what's significant, what remains after correction of multiple testing. We always are looking at p-value maps. Very little attention is afforded to effect size. They remain incredibly important effect sizes for our understanding of how the brain works, and of course, for understanding pathology. Not only the direction of effect is important, or of effect size is important, but also the impact of confounding and residual confounding on effect sizes is important to understand. So what is confounding? I'm not going to spend a lot of time on these basic definitions, as it's already been discussed by both Henning and Tonya, and everyone can visit Wikipedia on their own. One piece that I do want to discuss right now that I would like to highlight is that confounding, in epidemiology at least, is treated as a causal concept. Well, what does this mean? If you're adjusting your analyses for a particular confounder, likely you're also interested in the magnitude of an effect estimate, or better said, the association between your predictor and outcome variables. If you weren't interested in the magnitude of the association, you wouldn't be adjusting. Miguel Hernan makes a very interesting argument that this fact alone, the fact that we're interested in the magnitude of the association between a predictor and an outcome, suggests that we're also interested in making causal inferences. Now, I'm going to leave it at that and suggest the C-word paper as more interesting background reading for you on this topic. But whether or not you agree that adjusting your models for confounders implies you're doing causal inference, these adjustments do have an impact on our statistical models. How is that? Well, the main way that we experience this often is to see an attenuation in an association between our predictor and outcome variable if a true confounder is present in the model. So for adjusting for a true confounding factor, we see, for example, a regression coefficient attenuate or get smaller. So any association we observe between our phenotype, so some kind of psychiatric condition, and our imaging data will be attenuated or reduced after we adjust for it. So just think of smaller regression coefficients as this example. However, given that our confounders are often measured with some error, some precision, lack of precision, we are left with something called residual confounding. Essentially, we're unable to fully adjust our models for confounding factor because we have not measured it with complete precision. When residual confounding is present, it's very likely that our association is still inflated and is an overestimate of any true association. So for modeling an association between a psychiatric condition and the brain, any effect estimate that we get, which is still confounded or has residual confounding, is likely an overestimation of the true association. If your model estimates, for example, beta coefficients, attenuate substantially after you've adjusted for confounders, it generally means two things. First, your association was or is confounded and that confounder is really a true confounder. If it's not a true confounder, you wouldn't see an attenuation in the association between your predictor and your outcome variables. Now, the second thing is, if you see a substantial attenuation, there is a high potential for residual confounding, as it is difficult to measure all of these confounders perfectly. An example of what confounding might look like in child psychiatric imaging, which is my field, is as follows. We often set up our models so that some underlying feature in the brain predicts psychopathology or symptomatology. Although Henning showed in the previous lecture that this setup is flawed given the potential for reverse causality, but I'm going to ignore that for now. If we think about prenatal exposure, such as cigarette smoking, there is evidence that features of the brain are related to exposure to tobacco. Further, several studies have also found associations, albeit somewhat weak, with prenatal exposure to tobacco and increased risk for ADHD later in life. Thus, prenatal exposure could explain any association we find between the brain and ADHD in a confounded fashion. Another clear example is with socioeconomic status, SES. Several lines of evidence exist showing that those exposed to disadvantaged backgrounds are at a higher risk for adverse health outcomes. Now, you can also imagine these constructs, prenatal exposures, socioeconomic status, act as confounding factors. There are potentially more unmeasured confounders or other, for example, other opportunities to measure these confounders, which would help us minimize the chance for residual confounding. But this all really depends on the phenotype under study and how confounded it actually is. For example, in nutrition research, it's often very difficult to adjust for all potential confounders as they're numerous and the potential for residual confounding is very high. If we think about whole brain analyses, so for example, voxel-wise analysis, vertex-wise analysis, think about TBSS, voxel-based morphometry, VBM, QDEC, and so on. We see a relatively simplistic approach to dealing with confounding. Actually, not just in these voxel-wise analyses, but in general in imaging, we adjust for a fairly limited set of confounders. Think about age, sex, and maybe site if we're doing large consortial work or working on samples such as the ABCD cohort. There are several reasons for this. We don't adjust for so many confounders. Historically, sample sizes have been fairly small, and we don't have the degrees of freedom for additional covariates, or maybe we're using matched case control designs to adjust, or maybe we just didn't collect these confounders. But with bigger and bigger samples, proper adjustment for confounding factors is crucial. Another point you may notice is that most software for conducting these kinds of high dimensional analyses requires us to have complete information on all of our variables. Thus, we're forced to run a complete case analysis and exclude any data with missingness or handle missing data in some other way, so a mean imputation. So, for example, if we somehow do not have information about socioeconomic status, or we don't have information about prenatal exposures on certain individuals, we're forced to exclude the entire individual, or we need to do something else, such as impute those missing values. Further, other limitations of very widely used software packages limit to what extent we can adjust for confounding factors. So what we see is there's always a balance between user-friendliness and functionality in these software packages. The more user-friendly the package is, the less options we have for adjusting for multiple confounding factors. Some may only allow for a certain number of factors to be added into the model, while others are very, very flexible, but they're very difficult to use. So really quickly, what are the implications of confounding on whole brain analyses? Well, we know confounders often attenuate effect estimates. This attenuation may render some associations non-significant, and thus not adjusting for confounders could give us a very misleading picture of the spatial pattern of associations that we see in our whole brain analyses. So to give an example of what I mean by this, comes from some work by a PhD trainee in Rotterdam, Lorenza Diaglio. Here, Lorenza used the Generation R study, a prospective population-based birth cohort. About 2,700 children were present in this study with usable MRI data But they also had data on attention problems from the child behavior checklist, which is a 100-item, in this case, maternal report on behavioral and emotional problems. Now, Lorenzo used T1-weighted images processed through FreeSurfer and fit a general model across the entire cortex, assessing the association between surface area and attention problems, and adjusted for various sets of confounding factors. Lorenzo fit all of these models at each vertex using the brain data from a package that was developed by a former PhD trainee at Erasmus. Now he's a postdoctoral fellow, Sander Lambley. This package is the QDEC-R package in R, which has been developed to accommodate large population-based cohort studies like Generation R, ABCD, UK Biobank, etc. It has several novel options which are not present in some existing software packages, including allowing us to impute missing data. What Lorenzo found was that this picture of the spatial pattern of association between the brain and ADHD changed depending on how many confounders were modeled. Now, Henning already showed this figure. What we're looking at is a color map of the clusters indicating the association between surface area and ADHD problems. The more yellow the color, the more confounders have been added to the model. Red is the basic model, yellow is the quote-unquote fully adjusted model. In these analyses, we have socioeconomic status, SES, maternal age, child gestational age, prenatal exposure to tobacco, maternal psychiatric problems, and child IQ. The arrows on the screen show the regions which disappear after adjusting for certain confounders. Thus, as examples, portion of the posterior cingulate as well as the frontal lobe disappear with certain confounders, namely socioeconomic status, but also other confounders. Now what you see on the screen is a bar chart for each model. So a basic model with age and sex, ethnicity, another model with socioeconomic status, another model with maternal age, gestational age, etc. Each bar represents how many vertices or how many data points are present in the spatial maps after adjusting models for a given confounder. What's striking about this is the size of the clusters associated between surface area and ADHD symptoms shrinks considerably after adjusting for example, SES. The cluster size there is less than 60% of its original size. So when we adjust for socioeconomic status, we lose about 40% of the vertices. So indeed, the picture here changes dramatically after adjusting for confounding. Now, previously we said that as we add confounders into our models, we expect the association between the brain and ADHD symptoms to attenuate or become weaker. The histogram here shows the percent change in the regression beta coefficient estimate from the basic model to the fully adjusted model for all of the vertices in the brain. So 160,000 points in the brain. So we subtracted the beta coefficients from model 6, the fully adjusted model. We subtracted the regression coefficients from model 1 from model 6, so the fully adjusted model and the minimally adjusted model, and we created a percent change map for the brain. Then we plotted the frequency of each beta change from the basic model to the fully adjusted model for all, as I said, 160,000 vertices in the brain. Now what you can see is the histogram is centered at about 17%. That means we see a roughly 17% attenuation in the beta coefficient after adjusting for several confounders. And this is really the driving factor behind the change in the spatial extent of the clusters that we're seeing. I'm not showing it here, but if you look at the standard errors, the other piece to the puzzle for calculating a p-value, they are much more stable, meaning that this loss in cluster size is really driven by an attenuation of effect estimate, which is being driven by confounding. One last note on confounding before we move on. It's not always necessary to adjust your models for confounding. It really depends on what your research question is. For example, oftentimes in prediction work, the goal is really obtaining the best prediction, and thus we don't care too much about confounding, unless of course that confounding is leading to poor generalizability of our models. I would recommend another paper from Miguel Hernan which goes into detail on 3 primary types of research that we do, at least the conceptualization of these 3 kinds of research, Miguel Henning, which can help us decide whether or not our work requires adjustment for confounders. It's a really good background reading. I think a substantial portion of the work presented at the OHBM is really ideological, but this paper is helpful in helping you decide what kind of classification to put your work into. The next topic that I want to discuss is selection bias. So simply put, selection bias occurs when your sample composition is no longer consistent with or really represents the original population of interest. It occurs as example with loss to follow-up. One of the most obvious examples here is death, but it can also be individuals who are no longer interested in participating. They may not have time anymore. Maybe they're too sick to come into the research center. Their schedules are too busy. Another area selection bias can occur is in selective recruitment or sampling bias. So the most familiar instance of this is recruitment from a university setting. Parents of children, students, etc. This group of individuals who may respond to ads for research studies at a university campus, this group is very different from the general population. An important note is this all really depends on the design of the study and what the original population of interest was. So if that population of interest was university students, there's no real issue with recruiting from a university. However, the data cannot be considered generalizable past this population. Some other examples here of some concrete examples of selection bias. Case control studies very often compare extreme groups. So let's say severely affected patients versus quote unquote supernormal controls. So very high SES, very high IQ relative to the patient group. We often see individuals from disadvantaged backgrounds dropping out or even not enrolling into our studies. Imagine a single parent working multiple jobs, may need to ride public transit to get to the research center. These folks have a bigger likelihood of either not joining your study in the first place or dropping out of the study as it progresses along. Lack of motivation and personality characteristics may also prevent folks from participating. Severe illness is another factor. So someone who spends a lot of time in the hospital already maybe doesn't want to come into your research study. They may prefer to stay at home and rest rather than coming again to the hospital for another appointment. The main take-home message here is sometimes the most interesting populations that you want to study are the ones which can be very difficult to recruit into your study, and we have to be aware of whether or not we have a selective effect here. So returning to this previous example that I showed before on ADHD and whole brain analyses of surface area, for that analysis, we have several covariates: socioeconomic status, income, education of the mother, psychiatric problems, IQ, etc. Some of these variables in our study, in the Generation R study, have missing information for certain individuals. So if you look at the table here, there's a column for missingness, and you can see 8% missingness on educational level of the mother. And you can also see 12% missingness on family income. And these are just a few clear examples. If we use multiple imputation to impute our covariates, we can maintain our full sample of 2,700 children that we started with. If we were to do, however, a list-wise exclusion based on all missing covariate data, we would lose almost 1,000 data sets down to a sample of 1,700. We call this a complete case analysis. This is important because a lot of voxel-wise or vertex-wise packages that we use in brain mapping do not allow for missingness. So we have to do something about the missingness. That's multiple imputation, mean imputation, whatever you think. Otherwise, we end up with a list-wise deletion, so a huge drop in sample size. What you then can see in this table are some very obvious selection effects. With ethnicity, more Dutch individuals are retained in this sample and fewer non-Western individuals are retained. Further, we see more highly educated individuals stay in the study, and those earning more income also stay into the study. This is very clear selection bias if these data sets are dropped. If I were to rerun our whole brain analyses that we talked about before, in this complete case sample of 1,700 children, so where we lost 1,000 children due to missingness, you now see the striking impact of this selection. Now this histogram again shows the percent change, this time beta coefficient from the full sample to the highly selected complete case sample. Upwards of 50% change in effect size, effect estimates across all vertices can result from this selection effect. Now this is not related to power, this is purely related to the effect size. This leads to dramatically different results for the association between ADHD and the brain. How do I address selection bias? The best answer is try to avoid it from the start. So if it's selective recruitment, really think carefully about the study design, the recruitment, and try to minimize the selection effects as much as possible. If it's about maintaining the subject pool and not having lost the follow-up, also think carefully about how you can keep your individuals interested and participating in your study. But of course, everyone knows this is very, very difficult to avoid. And luckily, some methods do exist to ascertain the level of these selection effects. One method is known as inverse probability weighting. Essentially, each data set in the sample receives a weight based on how under or overrepresented he or she is compared to the source population. So for example, in the previous table I showed, fewer folks earning low income were retained in the complete case analysis. In inverse probability weighting, I would apply a higher weight to any data I have from individuals with low family income. This would upweight those individuals in any statistical analyses I have to combat these problems with selection bias. Now, these weights can very easily be passed to several linear regression tools, for example, in R. If your results change substantially after this weighting, you have a good idea that the selection effects are partially influencing your results. So if you see a big change after applying this inverse probability weighting, you know that the selection effects in your sample are a cause for concern. On the previous slide, I showed the impact of losing data due to missing covariate or confounder information. This was a bit of an extreme example, but it illustrates the importance. In epidemiology, it's common practice to impute missing confounder data, and sometimes even imputing missing outcome and predictor data, but that's beyond the scope of this discussion. In any case, this can be done with several packages, for example, mice or Amelia in R. This method of multiple imputation differs from mean or median imputation by making predictions of missing values based on all available data that you have, so within and between subjects. Essentially, you're running a regression to fit and make these predictions using several pieces of available information. So you may have some data available on one person, but are missing important covariate data on that person. And you can use all of the available data that you have to make the best, most informed prediction about what their missing values are. Often with multiple imputation, a distribution of predictions is built, say 30 imputed data sets. Each imputed data set is used to run a given analysis, for example, a linear regression. And then the results of the regression are pooled across imputed runs. This methodology has been shown to vastly outperform mean or median imputation and is definitely preferred in epidemiology. One important note here is that we are mixing two concepts on this slide. One is confounding and the other is selection bias. So imputing data allows us to retain our full sample, which is minimizing our selection bias, and also allows us to adjust for several confounders, which is helping us mitigate confounding effects. The last point that I want to talk about today is adjusting our analyses for a global effect. So if you think about your volumetric analyses, it's very common to adjust for total brain volume or, for example, ICV. The basic idea here is specificity, of course. We assume that the brain areas which are related to our phenotype above and beyond any particular global effect from total brain volume are of the utmost importance and play a defining role for a given phenotype, right? However, I want to caution this interpretation just a bit. what does it mean if we don't see any regions related to our phenotype after adjusting for total brain volume or some global metric if we're talking about DTI, for example? Another example to illustrate this point and why I think we should be a bit cautious is from a paper by Martina Hoogman in the American Journal of Psychiatry looking at ADHD symptoms and, in this case, total surface area. Highly significant association between ADHD symptoms and total surface area, and thus we sort of assume that there is at least some component of a global effect here in ADHD. And when we test specific subregions or focal regions, we want to know if there are any of those that are more important or more specific than this particular global effect. Now, once again, I'm coming back to this same figure that I've shown several times now, and this analysis that you see on the screen is not actually adjusted for total surface area. It's only adjusted for the basic model age, sex, ethnicity, socioeconomic status, maternal age, gestational age of the child, any exposures prenatally, maternal psychopathology, and IQ. So what would happen to this surface-based map if we adjusted for total surface area? Many of you probably guessed, but all of the results disappear. So what does this mean? Does this mean that nothing in the brain is related to ADHD? With a somewhat haphazard analysis, that could have been an unfortunate interpretation of these data, especially if total surface area was put into the model and then never explored without total surface area added to the model. But so what exactly is going on? We know ADHD is related to the brain, and we've shown that there is some kind of global effect of total surface area. So where is that global effect coming from? Especially if we see in this figure that there are certain regions that seem to be particularly important. I think there are two equally valid interpretations here. The first is that this global effect is actually comprised of the subset of focal regions you see in this figure. So it's not a true global effect in the sense that the entire brain is implicated, but rather it appears to be a global effect when only examining a single variable, total surface area. The different regions highlighted in the figure then are sort of aggregated together and appear to be a global effect. And then, of course, if we adjust for this global effect, it causes all of these clusters in the brain to disappear. So that's the first possible interpretation. The second interpretation is there really is a true global effect, a small effect size throughout the brain with some focal areas showing slightly larger effect sizes that do survive multiple testing correction, but these small effect sizes throughout the brain do not, they fail to survive multiple testing correction. So in other words, a truly global effect. There's no clear answer here as to which interpretation is correct. However, I think both should be considered. Otherwise, we risk overlooking important areas in the brain, which may be related to our phenotype, but we discount or we disregard them because we're just always blindly adjusting for a global effect and not testing it without the global effect. The main conclusion here is be clear and understand why you're adjusting for a global effect. Check analyses with and without adjusting for a global effect. So for example, total brain volume, in order to detect a pattern such as the one that I presented today. If the results disappear, don't jump to a conclusion and automatically discount their importance. They may drive a global effect. In general, it's better to consider the pattern of the results as a whole instead of thinking about it as just specific or not specific, global or not. So in summary, I hope today that I've convinced you that adjustment for confounding is a crucial step in population level analyses. Also that results from whole brain analyses can change substantially depending on how confounders are treated. Often, again, we see our results shift toward the null after we adjust for confounders. Selection bias is a very serious concern and results can be biased in either direction. So you can either see an over or an underestimate of the true effect depending on the type of bias. There are methods in place for handling selection bias, such as inverse probability weighting. Lastly, I hope I've convinced you that at least to take a second look at adjusting only for global metrics such as total brain volume. Not doing so risks us missing important clues about how the brain is involved in various phenotypes. Thank you for your time. My email is at the bottom of the slide for anyone who has questions or would like to further discuss any of the topics presented here today. Thank you.